Okay, I think we can get started, even if people will still trickle in, because my introduction is not the most important. That's not why you're here. But good morning, good afternoon, and good evening, wherever you are joining for. It is my pleasure to today welcome you to the second day of ICCVAM's Method to Developers Forum. My name is Helena Hoberg, and I am the Deputy Director of the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICEATM. And I will start with just giving you a few minutes of background and housekeeping before we start our interesting methods developers presentations for today. First, I just want to apologize for yesterday. We had I know some of you had some issues for joining us. We think we have solved it for today, but in case you do not see any slides, please let us know in the Q&A as soon as possible. We have a backup plan for you if that's going to happen. But as I said, I really think we should have this working properly today. Next slide. So most of you are hopefully familiar with NICEATM and ICCVAM. ICCVAM stands for the Interagency Coordinating Committee for the Validation of Alternative Methods. But for those of you that are not familiar, or if you need a reminder, ICCVAM was established in the 2000 by the ICCVAM Authority Act, and it is composed of representatives from the U.S. Federal Regulatory and Research Agency, and the numbers of members have actually increased over the years, or the last years, and today we are 18 members. So these regulatory and research agencies require, use, generate, or disseminate toxicological and safety testing information. So what ICCVAM does, we conduct technical evaluations of new, revised, and alternative safety testing methods and integrated testing strategies with a regulatory applicability. But we also promote the scientific validation and regulatory acceptance or qualification of testing methods to accurately assess the chemical safety and hazards of relevant products. And we're doing this in a 3 Rs fashion to try to reduce, refine, or replace animal tests when possible. And NICEATM is we are the ones that provide the technical and scientific support to ICCVAM. And you can see our 18 members here on this slide. Next slide. So in March of this year, so 2024, we published a report that is called Validation, Qualification, and Regulatory Acceptance of New Approach Methodologies, which can help both developers and end users and regulatory agencies to build confidence in NAMs so they can be implemented to supplement or replace animal testing for regulatory and non-regulatory purposes, though we focus mainly on the regulatory purposes here. So you might be familiar with the ICCVAM strategic roadmap, and one of the strategic and important goal in that roadmap is to connect methods developers with the end users. And both the roadmap and this validation report are available on our webpage, and I really recommend you to go and read it if you haven't read it already. So what we're here for today is the Methods Developers Forum, and that was a natural follow-up activity on this validation report that you can see here on the slide, because it provides an opportunity for NAMS developers to present their methods and discuss regulatory issues with relevant stakeholders that are here today. So before this, on our webpage, we have regulatory and industry stakeholders. They were invited to record some presentations that in detail describe the specific needs and on evaluating the type of toxicity, and you can find those on our webpage. So we plan to have this Methods to Developers Forum twice per year, and each time it will focus on a specific endpoint of toxicity. And this is the first in a series then, and we had decided to focus on carcinogenicity, and we have selected methods developers that will discuss their methods and also how they align with the key concepts and the flexibility for fit for purpose NAMS validation that is described in this report. And you can see those on this picture here, like biological relevance, technical characterization, and so on. So you will hear all the methods developers are going to describe how their methods actually apply to these concepts. Next slide. So we have a disclaimer because this forum is intended to facilitate open engagement and scientific dialogue regarding NAMs for, in this case, carcinogenicity testing. And just because you're participating in this event, it does not imply that it's a regulatory acceptance or consideration of that method as a part of regulatory decision-making framework. So this is just an opportunity to share the scientific background about the methods. Next slide. So this is the agenda for today. We have 5 different speakers and we have some breaks included as well. So it's going to be a packed day with a lot of interesting presentations and information. Next slide. So we, of course, need to acknowledge a lot of people. I think I've done the least with this, but we have a steering committee that has been going over all the slides and make sure that the presenters are really presenting what we need to hear, and that they've also been working on how this event should look like. We have a lot of support, NICEATM support from our contractors, Innotiv, and I especially want to thank Ingrid Daniel, and I'm going to shortly hand over to her. First, I just want to give acknowledgement also to our AV support, Nathan, Paris, Chris, and John, and also our capturer, Amy, for today. So with that, I want to hand over to Ingrid Daniel that have actually done most of the work on this, and let her do the housekeeping and introduce our speakers. And I'm going to sit back and listen to a fantastic line of speakers. Thank you.
Thank you so much, Helena. I am a senior toxicologist supporting the NICEATM contract. And as Helena said, I've been helping to facilitate the Method Developers Forum. I'm really excited to be back for day 2 of this event. And we had some very robust discussion yesterday, and I'm looking forward to the same today. So if we can go to the next slide, I'm just going to go through a few housekeeping. I know most of you should be familiar with Zoom by now, but at the bottom of your screen, you should be able to find the controls that you will be able to use to interact with the speakers today. If you're not seeing it, please hover your mouse at the bottom and hopefully it will come up for you. We do ask that you rename yourself in the participants window with your first and last name and affiliation so that we can call your name appropriately if you have a question to ask. Next slide, please. So regarding questions, we do encourage you to use the raise hand function and ask your questions to the speakers verbally. We will do that as time allows. And then if we need, since we have a very packed agenda, if we run short of time, we will ask that you put your remaining questions in the Q&A written, and then the speakers can answer your questions as we move on to the next speaker. The next slide. Lastly, I just want to mention that this webinar is being recorded, and we will have the video and the downloadable presentations posted for you on the Method Developers website where the registration was. It's right there for you. And that, I believe that's the last slide. So with that, it is my pleasure to introduce our first speaker, Dr. Ellen Berg from AltoPredict, who's going to be discussing the Biomat Assay Panel for Test Agent Prioritization. So welcome, Ellen. And if we can bring Ellen's slides up.
Yeah, so thank you, Amber, Helena, the NICEATM organizers for inviting us. Our method is a collaboration between me at AltoPredict and Eurofins Discovery. So I'd like to acknowledge the Eurofins team. Thank you. So let's jump right into the method description. We are proposing a 3-assay panel for characterizing chemicals and test agents for immunosuppressive mechanisms. We believe that immunosuppression is a neglected mechanism in carcinogenicity risk assessment and for protecting human health, because humans are very different than our rodent friends in having evolved and educated our immune systems to manage very high levels of genotoxic exposures due to our much longer lifespans, the complexity of our environments, including decades-long exposures to UV radiation. So the panel of 3 assays are run in sequence. There is an endothelial cell cytotoxicity assay that identifies mechanisms associated with acute toxicity, and then an endothelial cell proliferation assay that identifies mechanisms associated with organ toxicity. And then finally, a T cell proliferation assay that identifies immune cell specific mechanisms that are associated with immunosuppression. This particular set of assays have been applied primarily in screening format for drug discovery, in early discovery, for hit triage, and for lead molecule characterization. And these 3 assays represent are members of a 148 assay commercial panel, the Biomap Diversity Plus panel at Eurofins Discovery. So this shows the detailed methods. These have been published down below. These are relatively short assays. They utilize human primary cell types and specific activation conditions that engage adaptive responses as indicated. And note that these 3 assays have been run for the EPA's ToxCast program, where they're known by the assay names listed. And this, oops, I'm trying to go back. Here we go. Here we go. And this is just to indicate that a number of carcinogens are active in these assays, and carcinogens that represent particular key characteristics here. There are more details on the mapping to AOPs in the supplemental at the end of the slides as well. So there are a number of advantages. The use of the early passage human primary cell types, these provide the highest level of physiological relevance for culture cells based on their ability to retain the normal regulatory pathways representative of cells in vivo. These assays represent key assays within defined toxicity signatures that had been developed using the entire panel for identifying acute toxicity, organ toxicity, and immunosuppression mechanisms, classifying agents for these particular risks. And they've been successfully standardized, scaled, and run under quality management systems for more than 15 years. And there's certainly a large body of historical data in the screening format for sure. Okay, so as I mentioned, these represent 3 of the 148 assays or of the large panel. The large panel, the Diversity Plus panel, has been run in screening format for a number of years, based on a large reference database. That the large panel has been used, profiling in the screening profiling in the large panel has been used to identify mechanism of action, and it's very good at classifying and distinguishing a large number of mechanisms, and in particular relevant to immunosuppression, glucocorticoid receptor agonists, JAK inhibitors, calcineurin inhibitors, mitochondrial inhibitors, mTOR inhibitors. These are all contribute to immunosuppressive mechanisms, can all be really well detected and distinguished across the full panel. So the choice to select the 3 assays is based on first on economics. These are provides an economical way to detect, the key mechanisms important for detecting immunosuppressive agents and distinguishing immune cell-specific mechanisms from those more general mechanisms of inhibition of general cell proliferation or cell death, cytotoxicity. In addition, the proposed new format is designed to be more statistically robust with 8 concentrations, multiple replicates within experiments, separate vehicle and key reference controls within experiments, permitting standalone statistical testing, so not having to rely on historical data for interpretation and confidence in the method. The number of limitations are, of course, vehicle and agent limitations typical for cultured cells. These systems do not have complete metabolic capacity, although they are activated systems, so they do have a different profile, a different pattern of metabolic activities than resting cell types. But certainly do not represent the full complement that would be provided by the liver, for example. And again, another limitation is on the historical reference data. Again, the assays in the past have been run in singlet. The data transformations actually incorporate the vehicle controls rather than keeping them separate within an experiment. And there's limited availability of full concentration response data, which would be required for determining, no effect levels, which would be important for any, any applications, regulatory, and so on and so forth. So the list of references is here that have data from this panel. And for the context of use, this is speculative because we are just now, developing this method for the potential use in regulatory context. It certainly has and could be used in prioritization and hazard ID in research, but it can help with MOA, certainly in the development of AOPs. Especially if we're starting to develop AOPs relevant to human outcomes, potentially point of departure, and definitely consideration as components for combinatorial NAMS approaches. So not on their own replacement of animal testing, but within a combination solution. The regulatory need, again, investigating mode of action, supporting weight of evidence. Regulatory space is pretty broad. The larger panel has been used in all of these areas, for various, prioritization or mechanism investigations. The data from the larger panels, I believe, certainly has been incorporated into regulatory submissions, typically to support mechanistic questions and decisions, not, to my knowledge, replace animal testing. But that we would not have privilege to know that information. Coming from at CROs, they often are not informed of where the data goes. So the regulatory agents would be better suited to answer this question. Okay, biological relevance. So mechanistic understanding, again, the assays have been mapped to mechanisms primarily based on the activity of reference compounds and drugs. See the publication list that was previously included. And we do have mapping of the assays to carcinogenesis, key characteristics, and AOPs. in the supplemental slides. And just to point out that these assays provide information that is far downstream of the molecular initiating events and early key events. They're closer to the AOP outcomes. They're downstream, and as a result, they tend not to overfit predictions. So if the compound or an agent flags in these assays, we find very high likelihood that there will be an in vivo effect if the concentration at the tissue site reaches an effective concentration. So that's a benefit, but how it fits into AOP development. Reference compounds. The key reference compounds for the 3 assays are listed here. There have been reference pharmaceuticals. There's a list that can be found at Eurofins Discovery that have been run, and profiles are available through Eurofins Discovery, and there is open access data in the CompTox dashboard for the 3 assays, and the links are here. And there's right now a number of chemicals that are in there, including some immunosuppressants as well. So comparison to existing laboratory animal methods, providing data on human mechanisms is, especially in, the immune biology is very important. The animal models just do not reflect the education of the immune system that humans have. And we've also discovered, because these 3 assays have been used in early discovery, that they're quite useful for identifying, determining starting in vivo concentrations. So even in animal testing, I mentioned the human primary cells. They're retain the regulatory mechanisms of cells in vivo, and this data is highly complementary to orthogonal information from other assays, chemical properties, and so on and so forth, that and we feel can help characterize and categorize these mechanisms. Okay, technical characterization. This panel has been used, was developed and has been run in a commercial setting for a number of years. So a full quality management system is in place. So there's been continuous tracking of experimental variables and assay performance metrics, for many years, which has allowed identification of sources of variability and investigation through typical Kappa processes when variations are discovered. And so there's good quality control of these assays. Methods to identify and mitigate cell contamination are in place. The endotoxin, for example, actually gives a signature in these assays, so it's actually pretty easy to detect contamination if it's somehow missed along the way. And because this has been used for the ToxCast program and many consumer products, applications, pharma, food materials have been tested. So the compatibility with a number of different materials has been determined. Robustness is typically determined by reproducibility of responses to the positive and negative controls. There's 2 types of controls: response to activation conditions, giving you an activation window, and then response to a reference control. There's a cutoff for %CV of vehicle controls and comparison to historical data. The assay acceptance criteria in the past has been based on the profile across multiple endpoints. And so with this new format, we will obviously be developing a different set of assay acceptance criteria that will be more fit for purpose. So intra-laboratory reproducibility. So here's an example of testing the reproducibility of the T-cell proliferation assay. This represents 8 independent experiments. As I mentioned, these are these are in singlicate, but so there's 8 experiments. This is the response to a positive control sirolimus and compared to the vehicle control. So there's this is how we assess intra-laboratory reproducibility, and then again could be used for interlaboratory. The assay platform has been transferred a couple of times, including across state lines with new technical staff and successful transfer assessed by meeting the quality management assay acceptance criteria, which has to do with meeting those quality metrics. So it's a pretty standard method. It has not been transferred outside other than this, and not in the format that we are proposing. So I just had a few supplemental slides. These are the assay methods. These are from publications. So details on the assay methods, the assay acceptance criteria, mapping to some relevant AOPs and key characteristics here. And I think that is all we have here. And I'd like to thank my collaborators at Eurofins Discovery, in particular Justin Lipner and Diane Worth, Alistair King, and the team there. And happy to take questions.
Thank you so much, Ellen. That was a very interesting presentation. If those who have questions for Ellen, please go ahead and raise your hands. In the meantime, I do see a couple that are written questions. The first, immunomodulation does not necessarily equate with a cancer hazard or risk, but broad immunosuppression is usually the red flag. Can this set of 3 assays distinguish between the two?
Absolutely. And I think we still have a lot of work to do here. And I think that's where we're super interested in building up the AOPs that connect. There's known drugs, immunosuppressive drugs, that result in, into increased incidence of particular cancer types. That's the area we think has been a little neglected, but these assays would be super helpful for.
Thank you. Next question. Clinically relevant immunosuppressive is certainly an important element in carcinogenicity. The tumor types induced in immunosuppressed patients are specific, typically due to cell proliferation induced by latent viral infections, as in lymphoma. This implies it is not those where the role of the immune system to control removal of initiated cells is compromised. Solid tumors are not elevated in immunosuppressed patients. Have the reference compounds been scrutinized for the induced tumor types and whether they match the oncological clinical pattern of tumors in immunosuppressed patients?
We're just exactly just starting to look at that. And it's going to be complicated because, as you know, patients are also taking a number of drugs. So it's a little bit of a whack-a-mole. It will take a lot of data and a lot of, connecting to the clinical data of individual patients, what drugs that they've been on, even, their, risks from other environmental exposures. So it has not been, fully done yet, and we're looking forward to investigating that.
Thank you. Next question, I see John Elliott has his hand raised.
Hey, everybody. John Elliott at NIST. Question about the cell lines. You're using, not cell lines, primary cells. You're using primary cells for your assays. Presumably there's donor to donor variability. There are all the issues that happen with primary cell lines. They don't always behave the same, et cetera, et cetera. What is the quality around that part of it look like? How do you normalize that out? Or you don't even have to necessarily explain it, but just have those concepts been addressed and kind of thinking through how to use cells? It might be from different donors in your assays.
Yes, and that is the secret sauce of that platform. The investigation that went into how do you manage donor variability. So all of these 3 assays use cells from multiple donors. They're pooled. In the case of the T cell proliferation, they're pooled just before assay. They are pre-tested for pre-activation. Occasionally, a donor lot will come through that's got an infection or somehow is different. The beauty of this technology is that it will detect that in the unactivated conditions. So there are systems set up to detect that. Yeah.
Great, thank you.
Great questions. Thank you all so much. I'm not seeing any other hands raised right now. If you have any other questions for Ellen, please go ahead and put those in the Q&A box. And thank you again so much, Ellen, for your presentation, for joining us.
Thank you.
So now we'll go ahead and move on to our next presentation. I'm happy to introduce Dr. Gina Hilton from PETA Science Consortium International and Dr. Amber Getz from Syngenta Crop Protection, who are going to be speaking about a reporting framework called RECAP.
Great, thank you so much, Amber. And just confirming, can you hear me okay?
Yes, thank you.
Awesome, thank you for that introduction. So my name is Gina. I'm gonna kick us off the first half of the presentation, and then I'm gonna hand it over to my co-presenter, Amber, to give some more technical details. So I'm gonna jump right into the presentation today, where we are going to be describing a reporting framework to support a weight of evidence safety assessment without long-term burden bioassays. Slide, please. And our outline, it follows the other presentations. So I'm going to give the method and outline context of use. And then Amber is going to walk you through a case study to illustrate biological relevance. And then we're going to wrap it up with some technical characterization and scientific validity. Slide, please. Thank you. And then one more time. Okay, so you're going to notice that one of these methods is not like the other, because our method, if you will, is actually more of a framework for data integration to help support structuring a weight of evidence safety assessment to protect against chronic risk. So this framework stems from a project called RECAP, and that's short for Rethinking Carcinogenicity Assessment for Agrochemicals Project. And I'm not going to spend time going into details about the concept of weight of evidence, because I'm thinking that you're all familiar with this within regulatory practice, but I do want to highlight that the goal of our data integration framework is to estimate a health protective point of departure for chronic risk, and that's including protecting against carcinogenicity. So the focus really of this method is going to be application within agrochemical risk assessment, and then we can certainly explore investigating application beyond agrochemical risk assessment. Slide, please. And a very quick bit of background for those of you who are not familiar, and I see that you can't really see what's on the slide here, but this is just to give an example of the typical studies to fulfill a human health risk assessment for a conventional active ingredient pesticide within the US. This red box here in the right-hand corner indicates the guidelines for chronic tox and CARC studies. And again, just for those not familiar with agrochemical toxicity testing and risk assessment, there's about 20 toxicological studies that are conducted to inform the human health risk assessment within the US. And so we're going to demonstrate how to use this recap framework to address data needs for the carcinogenicity assessment. Slide. And what you're going to notice is that the development of this framework was really inspired by the ICHS 1B addendum for pharmaceuticals that we heard about yesterday in that presentation from the FDA. I'm sure you've all, maybe some of you have heard other presentations on this topic. And I just want to point out a couple of key differences here in the bolded text between the information that's considered between the S1B addendum for pharmaceuticals versus the recap framework that we started off for agrochemicals. That for recap, we're focusing more on read across and then also intended use of the pesticide, potential exposure scenarios, and then integrating all of that information to inform an estimation for a point of departure for chronic risk. Slide, please. And I'm going to briefly cover the method workflow for recap, and then Amber is going to show us the details, so the more fun stuff. So the framework starts with an evaluation of the active ingredient pesticidal chemical class to assess if there are structural analogs available. And then next, this is followed by genotoxicity assessment of both the target and the chemical analogs, followed by an assessment of potential non-genotoxic mode of action. Again, this includes an analog assessment for potential modes of action, followed by ADME, so thinking about TK and PBK modeling. And you're going to notice that the read across assessment is covering every single aspect of the toxicological information that's available. So really, we're trying to leverage read across at every single stage where that's available. Slide, please. And then once all of the relevant information is gathered and data can then be interpreted to elucidate a toxicity profile, and a point of departure can be estimated to protect for chronic risk, again, including protection for carcinogenicity. And now that we've had this background for a method, we're going to take a look at the context of use of this workflow with regulatory implications. So slide, please. And then one more time. Okay, so the context of use for this method, this framework to be integrating data, is associated with agrochemical risk assessment. Specifically, we're going to key in here for exposure assessment. And the example that we're going to work through today is in dietary exposure from food and water. And you'll see here that there are other exposure routes that could potentially be of concern, like residential or occupational exposure. But we're just going to keep it simple today and look at food and water. One important concept related to EPA's chronic risk assessment is the calculation of the CPAD, which is the chronic population adjusted dose. And this is the dose at which a person could be exposed over the course of a lifetime with no expected adverse health effects. Slide, please. Thank you. And so let's on CPAD for a minute, because I promise this is important. The context of the regulatory application here that we're proposing, we know that the EPA and other regulatory authorities are currently requiring chronic risk to establish. So the carcinogenicity and chronic studies can be used to establish the NOEL that's used for the point of departure in calculating the CPAD. So this is just one line of evidence, taking either the chronic study or the carcinogenicity study to calculate the CPAD. And what we are ultimately trying to do with the recap framework is to integrate data as a weight of evidence to estimate a health protective point of departure that can be used by an agency to calculate a CPAD without relying on the rodent cancer bioassays or chronic tests. Okay, so slide 12, we're going to look at the next slide. Sorry, we're going to look at a quick example to understand chronic risk assessment for those who aren't familiar. Here you can see in the blue box, sorry, that's hard to see. I should have made that red. Here, this is chronic dietary for all populations. This is an example of a human health risk assessment that used the mouse carcinogenicity study to calculate the percent CPAD. And then the next slide, this looks at the context of use, where the percent CPAD, the context of use for the US EPA in calculating the percent CPAD, it has to be below their level of concern, which is 100. So the CPAD is calculated by dividing the dietary exposure, which is estimated using the DEEM model, by the CPAD. And so we're looking at the percent CPAD here in that blue text for the most sensitive population. And this is typically what's used just to be conservative in that chronic risk assessment. Okay, so next slide, please. Okay, so I just said a huge amount of words, and really, so if you tuned out, this is a good time to tune back in. At the end of the day, this is what we're trying to do with the framework. So we are trying to look at how to integrate all of these lines of evidence, where here's a comparison of a CPAD that was estimated using the recap framework, looking at these multiple lines of evidence, including read across, metabolism, additional mode of action information, and then comparing this to what EPA initially had generated as a CPAD using only one line of evidence from the example I showed from the mouse carcinogenicity study. And so in this specific example, the recap estimate was more conservative in its protection, but it is important to note that both of these CPADs were below EPA's level of concern of 100%. Okay, and then slides. So just one more note from me. I do think it's important to land on this, thinking about context of use, that there is already an opportunity within the US to use this type of weight of evidence method. So we're in a position to be able to leverage this approach to support a chronic risk assessment for pesticides within the US. And really importantly, I think that there's an opportunity to expand what we've done for agrochemicals to other products and types of safety assessment, for example, industrial chemicals. Okay, so that is it for me. Slide, please. I'm going to hand it over to my co-presenter, Dr. Amber Getz from Syngenta Crop Protection.
All right, thank you, Gina, and thank you for talking and showing the method, the workflow here. So now, as we do, it's quiz time. Let's see if this works. So I'd like to now, let's work through this workflow with an example to show how the results from the read-across analysis were incorporated into the weight of evidence assessment. At the end, we will compare the results of the estimated point of departure with the recent regulatory review of this active ingredient. Next slide, please. So we're going to be picking on one of our new acetyl-CoA carboxylase. This is what I'm going to call the nickname here shortly. Acetyl-CoA carboxylase inhibitor insecticide. I'm going to call it ACCAs for short from here on out. And this insecticide is proposed for use on foliar applications on crops such as soybeans and potatoes. So what I'll do is I'll walk you through this workflow and then we'll see how this compares. Okay, next slide, please. Thank you. The workflow for this approach is presented on the left side, as you'll see here. We began this assessment with work to understand the class of chemistry that we're working on and learn what we can from other chemicals in this family. For example, what are the type 1 and type 2 toxicities reported for these chemicals in this class, if any? Each parameter in this weight of evidence assessment provides evidence to identify significant effects and characterize that hazard for that target substance, in this case, our new insecticide. As these lines of evidence are compiled, as you're pulling in all the information that you're seeing here on the left-hand side, their relevance for carcinogenicity assessment and reliability in their quality, how good is this data, are also assessed. So as we look at all this available data that's generated on the source analogs that are used to assess the safety of our target substance, we will also look at all the available data on the target substance as well, with the exception of the long-term and carcinogenicity studies. Just a spoiler alert, that complete data package was generated for this new active, but we conducted this assessment while those cancer bioassays were being completed. So we did not have the final results, which was useful for us to put ourselves in that position saying, does this work? Are we going to get that similar result? So we'll show that at the end here. Okay, next slide. So following, oops, sorry. Thank you. So we can move on to the next slide here, and we'll just continue down through the workflow here. So following this assessment, we took a look at, as we mentioned on the previous slide, the genotoxicity and the mode of action for the target substance. We also took a closer look at the pharmacokinetic properties. They were reviewed to look for any sex differences in absorption, distribution, metabolism, clearance. Was there any sex-dependent change that would influence how you interpret the results, not only of your target substance, but as well as your source analogs at the same time? The next step, after looking at this preliminary review of, thank you, the next step of looking at this, what we know about our target substance, is now time to select those source analogs to conduct that read-across analysis. So the underlying assumption here is that we are using the similar property principle, and which states that molecules which are structurally similar are also likely to have similar activities. So the initial search for this potential read-across analog began with the pesticidal class of chemistry. And this is simply because chemicals that are known to have the same pesticide mode of action will have some commonalities in their structures and their toxicity profiles. So this was also expanded, and we took a further look to at databases such as PubChem and CompTox dashboards. Several of you, I'm sure, are familiar with those, if not curated them yourselves. I know a number of folks here. So in this case example, the molecular fingerprints that we used to compare the structural similarities, these were created using ToxPrints, Tanimoto scores, coefficients, as well as the 729 chemotypes. These are substructural features that are relevant to assess the toxicity of these chemicals. So this was really helpful for us. And you can see down on the lower left here, I compared this to a PCA plot, but this is to grouping and categorizing how similar are these chemistries within this class of chemistry. And ours that we're looking at here is a little dark spot you may not be able to notice there, right in that green circle, and it categorizes quite well with the tetronic and tetramic acid derivatives, as well as the DENS. As you can tell, I'm not great with pronunciations, but I will try. That's the phenylpyrazolines. And you'll see that it is structurally different from the FOPs and the DENS. So looking at all of those chemicals that have a similar pesticide mode of action, we look at that structural similarity, but we don't start excluding the active substances and the analogs, excuse me, straight away. We keep them in there and we begin to take it to that next step. This is where we bring in the bioactivity. What are the biological effects of these? I want to look at those toxicity profiles and see what are those similarities and differences. Is there anything that can distinguish these? One thing that we did notice is that with the FOPs, these are peroxisome proliferators. And with our target substance, we did not see such evidence of peroxisome proliferation. So we felt that those could be excluded, not as reliable to compare the bioactivity profiles. So again, you also notice, as I mentioned, that the phenylpyrazoline that's in there, that did categorize with the tetronic and tetremic acids based on our ToxPrints fingerprints model that we used. However, this substance was excluded ultimately from the read across. This was based on the comparison of the bioactivity of the profiles. We noticed there was a difference in the target organs based on our available data that we had in the short-term and subchronic studies, as well as the endpoints used in risk assessment. These were not so much, well, they were not related to carcinogenicity. What I mean by that is this den did not present with any evidence of carcinogenicity, so that reduced our concern for excluding this from this read across analysis. Keeping in mind the context of the use for this read across. Had we been using this for a different endpoint, we may have kept that in there. So we're already starting to segue into the technical characterization and the uncertainties that you do need to consider. But for the case of this, for the purpose of this case study, we did exclude the DENs and we focused on the TAs, the tetronic acids. Next slide, please. With all the available data now collected and reviewed, comparison of the data packages were reviewed for consistencies across the analogs. Were there effects on the thyroid? Was there evidence for immunosuppression? I had a very nice presentation of that just now. Were there precursor findings in the subchronic studies that could, if you will, forecast the findings recorded in the carcinogenicity studies of the source analogs? Do we see some patterns here? What are we noticing about this class of chemistry giving us that information? How useful, what's the quality of that data that you're using, and how well can we rely on that in our weight of evidence assessment? While there were effects that were recorded with the source analogs, they were not observed with our target substance. We did see precursor findings that were recorded in the subchronic studies with those analogs, and we were able to, if you will, trace those effects throughout the data package. So the absence of such findings with our target substance reduced the likelihood of this tumorigenic response in the long-term studies. So again, based on the read-across analysis and comparing kinetics became very important to us, as we noticed, to really help us to understand the similarities and differences across the chemistries. The concern for the carcinogenic response to the target substance was considered to be low. The mode of action research on the thyroid demonstrated a non-human relevance and a clear threshold for this effect. At the end, we identified the dog as the most sensitive species, and its subchronic NOEL was identified as a suitable point of departure for the chronic dietary risk assessment. Okay, so pencils down, time to pass your test in. Next slide, please. Did we pass? So here's a side-by-side comparison of a recent regulatory review of this active substance for its import tolerance. So for an import tolerance, this is not including, as Gina described, both dietary and drinking water. This is looking at dietary. I did anyway take a look at that because it is currently under regulatory review for domestic registration as well. But what we're looking at here right now is the data package that these reviewers were presented with was a full data package, both cancer bioassays, as well as the chronic study in the dogs. And using this recap framework, this weight of evidence assessment, using the read across, you'll see, again, we're seeing a theme here, is more conservative. And we did include an additional uncertainty factor for the extrapolation and for the uncertainties. And with these approaches, they're both well below that level of concern. And we did actually select the same endpoint. So that was very encouraging to see how well this framework is working. Okay, so I'm going to keep us on time here. So next slide, please. Let's take a look at those uncertainties here. One more slide, please. Thank you. So there are uncertainties with this approach, and these must be addressed, including the quality of the data used, as I've mentioned, the change in interpretation of the data packages from one region to the next. Where is this going to be submitted? You need to focus on the regional jurisdictions, governing bodies, and their interpretations of the data package, protocols that they use. As well as the ability to use this approach for data poor chemicals. What we've mentioned here with the ACCAs, we consider this to be a very regulatory relevant, data rich mode of action. So there's a lot to work with. Okay, so data quality, how reliable are these lines of evidence that are used? That takes us right into the robustness. So what we did is we took a look at, we did not do the interpretations ourselves. We relied solely on the regulatory agencies. We selected one agency and their registration reviews of all of these analogs that we looked at. We wanted to use that consistent approach in how the assessment and interpretation and decisions were made. So based on that, we were able to reduce that variability. However, through these reiterations, as you can see here, this has been a multi-year process. We do recommend that you do look at all available regulatory reviews to look for those inconsistencies or perhaps distinct differences, because those do need to be addressed. Reproducibility. This case example used a target substance which does have a regulatory relevant, as I said, data-rich mode of action. Those that may have maybe data poor, limited data, or there may not be a very large number of analogs to work with. This may not work for those data poor chemicals. However, what this assessment will do is it will determine whether or not there is sufficient knowledge, there is sufficient data, and identify those gaps that might require more work. So it's not a guarantee that it will work, but there's also a good tool to use to determine that in the first place. And as we mentioned in other talks earlier today and yesterday, the transferability. How well does this framework work with other regions? There are efforts underway as we speak, and this method has recently been approved by the OECD ICCVAM Forum, Integrated Approaches to Testing and Assessment. And we are, and this is going to put our hands out there, is that we are on the search for additional case studies to better understand how well this approach does work. So we will be throwing our emails up there, and we do encourage you to reach out. So before we wrap up here, a short overview of this iterative process. As you can see here over the time, it really has been an iterative approach from outlining what are the data needs. Then we're going to evaluate this framework by preparing these case studies, one that you just saw, and getting those review of these case studies, not only from our work group collaborators, and just a big shout out to everybody there on the lower right. We could not have done this without such great collaboration and teamwork throughout. So we've had both our work group collaborators as well as regulatory reviews taking a look at these and providing that feedback. So over these past few years, 2-3 years, lots of revision to improve and progress this going forward. So a big thank you to everyone. And finally, I promise I'm almost done. One more slide. And this is just to share, as we mentioned, that working with the OECD recently over these past year and a half, 2 years has been really ideal. It's helped us to review the applicability of this case study for data integrations. And so we've had over 8 regulatory authorities provide hundreds of hours of review time on this. So it's been really helpful. And top 3 things that we got from the feedback is 1, ensure the broad applicability of this framework for this data integration. Take this beyond the pesticides, take this on. Let's look at industrial use as well. Number 2, provide a sufficient level of detail. As you mentioned, there are some uncertainties, so you need to provide that narrative, what was done, why was this done, and provide that justification of what was included and excluded and why. And 3rd, adequately capture and report those uncertainties so that we can help to understand how you interpreted that data, so we can have those discussions. So overall, there's unanimous support for this moving forward. And we do encourage folks who are interested to reach out to us. Last slide, just again, one more thank you. And thank you for your time today. And we do any opportunity to share our work. So we appreciate your time today and are happy to take any questions. Thank you.
Thank you so much. That's an incredible amount of work and a very nice presentation. And thank you both so much for sharing. What questions do we have for this presentation?
And if I can just jump in really quickly while we wait for
Sure.
a question, I am very inspired by the presentations that we've heard over the past couple of days, and also even beyond what's been presented today. And it is important to note that the framework that we're proposing here, it's a starting place. So agrochemicals, there are a lot of data requirements, as I showed early in the presentation. And so the goal ultimately, while we were estimating point of departure with traditional toxicity studies, I think the goal really is to be trying to have better data integration using these new in vitro and silico non-animal methods. So we're working towards improving our mechanistic understanding. And I know that OECD under the WNT, as we heard yesterday, that's a big interest on their side through more of that AOP structuring of data integration. So this is where we are. And I think that we're, I'm really inspired to see where the field is going.
Thank you. Not seeing any questions come up. Oh, there's one. Great. Question from William Bisson says, great case. I confirm the relevance of increasing the applicability domain, especially in the area of non-genotoxicity, considering we have polypharmacology and biological similarity from multiple levels. So more of a comment, I'm sorry. And then another question, if recap yields more conservative estimates of risk than current methods, wouldn't it be viewed as overly conservative?
Yeah, that's a great question, great point. And it's something we've certainly discussed with regulatory authorities, even beyond the EPA. And I think that this is a topic that we'll continue trying to work out and trying to investigate. Some of this comes down to, and we didn't talk about this today, but this really comes down to uncertainty factors. So had we not applied additional uncertainty factors in the examples that we used between a subchronic and a chronic study for estimating point of departure, it would have been comparable to what EPA had initially proposed. Mm-hmm. So it's, I can't speak on behalf of the EPA, but it is something that we're aware of, and we're conscious that we need to toe that line for it to actually be used. Then, that's something we're going to have to consider. I don't know, Amber, if you had additional thoughts.
Just to add to that, yes. So we have been, we've had another recent, it's a case study in the making, and using our benchmark dose, I can't talk today, apologies, benchmark dose modeling in our transcriptomic study. And afterwards we compare it with our conventional data package, and it's off by 2 milligrams per kilogram body weight as that endpoint. So it is very close. So let's work on our kinetics, let's work on our mechanistic and our mode of action, our understanding, and the more that we can provide in that area, the closer we make that bridge, and we reduce that gap. So you're absolutely right. It is very conservative to start with, but we're making efforts to get a better, let's close that gap.
Yeah, better certainty. Good question.
Great, thank you so much. I see one more. What are the plans to handle data quality, especially for key assays that have dose response curves, mechanisms, etc.?
Yes, so that's a question that has come up during some of these reviews that they've been through, and it is, how do you, are you just going to use any data that's available out there? How do you know that data is not is more distracting than relevant? And that's where we're looking, working on for us, where we started with was regulatory reviews. So if you will, we're using what I consider very reliable reviews, looking at that data. Once we start bringing in, as you say, more assays, more non-guideline studies, that's going to be another layer that we'll have to discuss. Interesting enough, we are working on another project on that right now. Happy to talk further on that. Is how do you incorporate additional non-traditional testing into your weight of evidence? So that's essentially underway right now.
And just really quickly, one more point on that, Amber. I think this is also the benefit where it is available to be leaning on additional analog information to help support proposed mode of action, if that makes sense.
Yeah, oh, sorry. Yes, I think interesting. I mean, the software is always important, and so I just yeah, no, great answer. Thank you.
Is there a way to establish a threshold for when a chemical class become data rich enough to use recap, so someone doesn't apply it inappropriately to data poor classes?
Yeah, this is something we've talked about extensively in the OECD forum. Exactly this point of how do you discern are 2 chemicals enough? Does it have to be 15 chemicals? Like, really, what is the threshold? I think where there is strong evidence of a supported mode of action. Well, okay, sort of two answers here. I think the more information, the greater the mechanistic understanding of a chemical. So when we first started investigating the development of this framework, we did have this discussion of what if this is a first-in-class scenario? So what if there are really no opportunity for a read across? Then some of the discussions that we had within our work group was essentially the compensation for that, of you needing to have greater mechanistic understanding. So they're not falling into that trap of we don't have enough information to support the toxicity mode of action. So I think that it could be applied for these classes of where there's not a lot of information, or maybe there's not read across available. It would just be, again, thinking about that weight of evidence, there would have to be other sources of compelling evidence to support. But I mean, it's really hard to answer because we just don't have, right now, we don't have guidance. And that's something that we're trying to work on within the OECD, is to better understand that tipping point of when is enough information, whether if it's for read across or PBPK modeling, what have you. And that's where, as Amber mentioned, having additional case studies is going to be really critical to demonstrate different use contexts. For example, industrial chemicals. Like, I would love to see the application of this framework to better flesh out more of those uncertainties.
Yeah. Likewise with genotoxic carcinogens, we've been focused again on agrochemical, which are 99.9% of the time non-genotoxic carcinogens, if carcinogens at all. So again, building that in there to really work through those unknowns. I'm really leaning on I have a better understanding of the mechanistic action, the mode of action that's happening, that's going to be helpful. Stephen, I do see your question there on how might disease susceptibility fit into this framework. Great question. Gonna have to have a think about that. Been again very focused on the carcinogenic potential of a chemical and how to address that, but we'll follow up on that.
All right, well, Gina and Amber, thank you both again so much for your participation in your presentation today. And thank you all for the great questions. Now we're going to go into a short 10-minute break, and we will reconvene at 10:20 Eastern time for our next presentation. Okay, it is 10:20. Hope everyone's feeling refreshed from the break. So now I'm very delighted to introduce our next speaker, Dr. Marc Audebert from the French National Institute for Agriculture, Food and Environment. And he is going to be telling us about a method for genotoxicity mode of action determination.
Hi everyone, thanks a lot Amber, and thanks ICCVAM to give me the opportunity to present the method named gamma-HDX phospho-H3 for genotox mode of action determination. For the method description, first of all, a brief recap of the importance of the genotoxicity assay for carcinogenic chemical risk assessment. As you know, the carcinogenicity assay spoiled by mice or rats costs a lot. It takes 2 years. It's not 3 or compatible, but they are regulatory, and there are some trouble with the predictivity because notably of the difference and interspecies between rodent and human. So a lot of different genotoxicity assays have been set up more than 40 years ago now with cell lines, and it costs very few. They are easy to perform in 2 weeks. There are 3R, and they are more and more acceptable. There are some test guidelines for some of them, and their predictivity are much more important now. And so because we are an increased need for in vitro testing in compliance with new legislation in Europe and US and all over the globe, it's very important to develop new assay for genotoxicity screening. So there are some limitations of the current use assay. First one is maybe the intraspecies difference between bacteria, a human, and rodent. We know the difference for metabolism, but also for DNA repair and for physiology. And then because there are much more compound on the market, we need to have high throughput screening possibility. It's not the case for some of the genotoxicity tests used at the moment. And also now we need for regulatory genotoxic mode of action. So we want not just to know if your compound is genotoxic or not genotoxic, we want also to know if the compound is an antigen, so it will induce a change in the number of chromosomes in the cells, or is a clastogen, so it will induce modification of the genome of the cells. And last but not least, now a lot of work have been done to determine point of departure. And so you need to have a very good dose response for your assay. So the method I want to introduce is, as you know, induction of DNA damage and mutation is strongly correlated with chemical carcinogenicity potential. We know that more than 90% of carcinogenic chemicals are genotoxic. Then the in vitro battery used at the moment has a low specificity, notably with false positive hits, especially with a mammalian cell base compared to in vivo. And so as explained before, we need now to have an understanding of the mode of genotoxic action, and notably in the idea of AOP. And so the method I want to show you have been developed for more than 10 years now and is based on histone, H2X, and H3. So histone are protein part of the nucleosome, and they are protein directly surrounding the DNA. So it provides a very important biological context and high relevance for toxicity endpoint characterizing genotoxicity and carcinogenicity. So the histone H2X could be a phosphorylated, named in this case gamma H2X, and is a very cellular response to DNA damage resulted from different DNA insults. So this biomarker is a well-known, recognized precancerous and cancerous biomarker in vivo and is used as such in human cancer research. Histone H3 could be phosphorylated at 710, and in this name is phospho-H3, during mitosis by ORA kinase to allow chromosome condensation and segregation. And so phospho-H3 is a biomarker of mitotic cells. So what we have demonstrated is that while an increase in gamma H6 is observed after treatment with clastogen, phospho-H3 induction is observed after exposure to antigen. So these two biomarkers allow an effective discrimination of clastogenic and antigenic chemicals. So this method is based on the quantification of these two biomarkers after cell exposure. And in parallel, you can measure cytotoxicity to discriminate misleading cytotoxic chemicals. And maybe importantly also, this biomarker, notably gamma H6, have been able to demonstrate the carcinogenic property of other carcinogen like radiation or bacteria producing colibactin or some human virus. So the advantage of this method. So first of all, this method is very important for predictivity of genotoxic potential. So now we have a database with more than 800 different compounds that have been tested, and we'll compare with classical in vitro assay like micronuclei vitro or EMS. And clearly, it seems that the method is more predictive with more specific and more sensitivity that are commonly used genotox assay. The other point is that this method is first to permit to discriminate mode of action between antigen and clastogen. You don't, with a single assay, you have the response you need. You don't need to use a CREST or other method after micronuclei induction. So this method can be used with different genotoxic mechanism of action. So with a lot of compounds that have been tested, we have tried to test all the mode of action of genotoxicity for clastogen and antigen. And this method is not influenced also by classical false positive genotoxic chemical that give rise to micronuclei or P53 inducer, for example. We can discuss this point because for gene related to gene expression, we have some false positivity for genotoxicity. And you can provide quantitative data because you can use this assay with different highly impressive multi-well plates, so you can have a lot of dose curve. So the other advantage is this method is faster than micronuclei vitro because you do not have a cycle completion. As you know, for macronuclei, you need that cell cycle to have the macronucleus. In this test, because gamma H6 is very early and fast response to DNA damage, few minutes after the damage, you can test this method with a timing very short at the end. This method has been applied to any cell type because keep in mind that histones are proteins that are one of the most important for the cells. And so they are present in all cell types, in all species, from all organs. And in a review of the method, we have seen that this method have been tested with a lot of cell line, but also with primary cells, and notably with classical cell line used for GenTox assay like TK6, FG2, or APRG, as mentioned by Les Ratio yesterday. So you can use also primary cells if you want, and maybe importantly, you can use different cell type with different metabolism capacity, like APRG, and also sometimes some cells without devoid of any metabolism. So if you compare these two type result in two cell line with two different bioactivation system, in this case, you can have a great view of if your compound or not need bioactivation for genotoxicity. And this method could be also performed in 3D or in suspension or in adherent monolayer. And you could have a high throughput format. And you can find very easily available antibody against these two biomarker, and there are different kits from different supplier, but also screen service for different zero for this method. So here is a table of comparison of the difference between the different classical GenTox assay I use. So I put the COMET, the macronuclei, reporter gene assay, and gamma-HDX 433. So as mentioned, gamma-HDX 433 can detect 1 to 10 breaks because in fact, when you have one break in a cell, at the end you have more than 2,000 phosphorylation events. So you have an amplification of the signal. In case of COMET, we know that we need 10 to 100 strand break to be detected as positivity. For the macronuclei, you need a cycle completion, and you just check for DNA in the cytoplanes. And also keep in mind that for gene reporter assay, like Tox Tracker or green screen or whatever, or it could be also the case for TGX-DDI biomarker, you don't directly check for the DNA damage. Gamma-HDX is clearly an in vivo cancer biomarker, and a lot of paper that clearly demonstrates that precancerous and cancerous cells in humans are positive for this biomarker due notably to genomic instability. And this is not always the case for the other biomarker. Also cell, because histone are present in all cell type, clearly you can use the cell line of your choice. It's also the case for COMET. For macronuclei, most of the study are performed in TK6 cells, so maybe much more one cell type. And for reporter gene assay, depends what cell type was used for the assay. For the high throughput route, we know that here in case of reporter gene assay or gamma-HDX, you can use a high format first route. For macronucleus also now you can use some high format, and there's some trouble with COMET for that. Concerning the metabolism, as explained yesterday by Les Ratio, it's very important to keep in mind that if we want to have a good prediction of a human effect of chemical, we should use human metabolism. So in case of gamma-HDX 433, because you can use human hepatic cell line like APRG or FG2 or PAH, you can have the metabolism, human metabolism take into account, like maybe for the COMET. In case of macronuclei or gene reporter, in most of cases, if you use TK6 cells or cells devoid of any metabolism, you should use S9 liver fraction from rodents. So you have the trouble with the difference in interspecies metabolism. For the genotoxicity mode of action, so discrimination between antigen and clastogen, this method is the only one to do that. And so we have seen that this assay have a very high predictivity, maybe more than 90% compared to the other. We know that for gene reporter, you have some drawback, notably for P53 inducer, or we know that some TGDDI genome are not able to pick up antigen. That's also the case for the comet that did not pick up antigen, but they did not also pick up some mechanism of action like interstron link or something like that. And for macronuclei, we know that in some cases you have a high level of false predictivity, false positive predictivity due to apoptosis, notably. So you have a very well reproducibility of this assay. And for the validation, so as you know, for the comet, there are some recommendation for in vivo comet, but not for in vitro. So you have a test guideline for macronuclei, and there are some discussion at the moment concerning reporter gene assay. And at the moment, we are working for the test guideline development for the method gamma-HTX phosphorylase. So here is some example of the work done in my lab with this method. So I'm working a lot on food contaminants. So I've screened a lot of different food contaminants, but I've also performed some work on the validation of the method with numerous model, chemical and saline, and notably APIG and other. So for the method description, so the high-stone H2X and H3 phosphorylation status synthesis can be easily quantified. You can use a specific antibody against these two biomarker, and then you can use different technique for the quantification of this biomarker. You can use flow cytometry, you can use Western blot, you can use high content analysis imaging platform, but you can, there are also some people using proteomic mass spectrometry. So this biomarker and the respective antibody are not protected by any patent or license. So many suppliers propose antibody. And so it permits that I say is really and widely available to everyone. So there are some limitations of this method. As for most of the in vitro assay, you could have some autofluorescence with some test chemical, but normally it's easy to recognize with proper control, and so you can subtract out autofluorescence. Then, as for the other in vitro assay, at the moment, it's not easy to evaluate gases, but a lot of work have been performed at the moment with new methods like ILE to test some gases for genotoxicity with this biomarker. Then, since gamma-HTX is expected to be in part from DNA replication or transcription blocking lesion induced by DNA damage. So it seems that cells in primitive states may be more appropriate to clearly determine the genotoxicity of a compound. And moreover, for phosphorylase signaling, so as remember, it's a mitotic cells biomarker, so cells must allow to reach mitosis at the end. And the only little drawback maybe is that this method is not able to pick up a very specific antigenic mechanism of action with aura kinase inhibiting activity, as this kinase has in fact kinase that phosphorylate phosphorylase 3. If you inhibit aura kinase, you will not see the effect on the phosphorylase 3. So in this case, you could have supplementary biomarker, and in most of cases, people use now high content analysis, so you could have a screen also for other biomarker like polyploidy. So the context of use, so this assay is used a lot at the moment for screening and early selection of candidates before entry into development by company in-house or a service provider. It's a case for pesticide, for pharmaceutical or cosmetic ingredient. So it can be applied as early GenTox screening in parallel or as an alternative to the mini-EMS or macronucleus tests in order to predict the outcome of the regulatory in vitro battery of genotoxicity test. This method could also use for follow-up testing on mechanistic approaches for candidate under development and market product, notably to follow up a positive result. If you have a positive result in EMS or in macronuclei, you can use this method to provide more mode of action, notably for antigen or clastogen discrimination for macronuclei positive compound. And then you have also mechanistic study and read across approach, as noted just before, for retesting a market chemical under REACH or EFSA and other substance. And you can determine benchmark doses and potency ranking. When you have a lot of different type of compound from the same family, you can have determination of potency ranking for genotoxicity with this assay. So for the regulatory testing, this method can contribute to mechanism-based animal-free consent assessments of chemicals. And we're sure that this method will be fit to a component of a set of test method, notably maybe in parallel with QSAR, and notably for a mode of action of the compound. And then more generally, we try to implement IATA for genotoxic carcinogen assessment. So for the regulatory space of the method, this method has been used to screen a lot of different type of class of chemical, agrochemical, pharmaceutical, cosmetic, food additive, nanomaterial, and so on. And we have made a chemical space analysis of the 800 different chemical already tested and published. And we confirm a full coverage of the chemical space. So this method has been used for regulatory submission. This method was proposed as a complementary genotoxicity assay in a weight of evidence approach by WHO, but also for SCCS, for European cosmetic, and also some EFSA evaluation. And this method is currently used by numerous pharmaceutical companies in-house, but also is proposed by different CRO as services. And since 2022, we work on a detailed review paper and a retrospective performance analysis at OECD, and that will be contribute, I hope, in a few years to test guideline. So for the biological relevance of the assay, so as mentioned at the beginning of my talk, this two biomarker, this histone, protein directly surrounding the DNA, so it provides a very important biological context and high relevance for toxicity endpoint for characterizing genotoxicity and carcinogenicity. And so because of the very high efficient DNA damage response, these two biomarkers are very effective for discrimination between the two modes of action. And so that's why we're working at the moment in the context of advanced outcome pathway for genotoxic carcinogen and the development and NIATA, as explained yesterday in numerous talk. And we think that clearly the mechanistic information given by this two biomarker can translate as a molecular editing event and cellular response that are activated upon chemical exposure to carcinogenic compound for human. So for the reference compound, as mentioned, a lot of compounds have been tested with this assay and have been published already. So here, the database at the end is more than 800 different chemicals, and as you can see, is well balanced with the different mode of action. You have you have antigen, you have a lot of clastogen with a lot of different mechanisms of action, and you have also, and maybe most importantly, a lot of non-genotoxic cytotoxic chemicals. And it's very important to see the accuracy of a method. And so the different chemical type that have been tested so far on the gene are very classical genotoxic compound like tubulin binder or kinase inhibitor. And for clastogen, you have a lot of different mechanisms of action that have been tested with this assay, like oxidative stress, blockade addict, topoisomerase inhibitor, intercross-link, NTP pool imbalance, nucleoside analog, and so on and so on and so on. And maybe more importantly is all the false positive compounds that have been tested to be sure of the accuracy of the assay, notably for apoptosis inducer, or for P53 inducer. Normally, all these compounds should be negative and are negative in this assay, but are not, notably for micronuclei or for some other type of assay. And so in the first review, we have published 3 years ago, or more than 3 years ago, we see a predictivity of more than 94% for genotoxicity. So compare with animal, the idea of this assay, because at the moment most of regulatory use in vivo data, but we think that in the future, part of the other assessments of novel chemical will be reliable on in vitro assay. And so this assay may contribute to the clear reduction of unnecessary animal testing, notably following false positive or false leading positive in vitro genotox assay. So with an important accuracy of the method aims to replace and reduce animal testing by improving the prediction and interpretation of the in vitro data. And we hope that the mechanistic insight we have with the mode of genotoxic mode of action, clastogen, allergen, may also contribute to a refinement of follow-up in vivo testing strategy if needed in some regulatory testing. And use a competent human cell, metabolic competent cell line like PIG or FG2, and coupling with this method permit to avoid the use of S9 liver rat extract. So and so yeah, and some read-across study also for sure. So for the technical, it will be very short at the end. So as mentioned in other point after, this method have been developed more than 10 years ago now, and have been extensively validated by Intel laboratories in different thermal levels with different technique of quantification in different lab in private or public labs. And different cell culture conditions have been used also. So cell in suspension like tecasic cells or adherent cell line like FG2 or whatever. And also now there are more and more publications with 3D spheroid models. So for the robustness, yeah, lot of inter lab and business, more than 90% of co-contacts between lab. For intra-laboratory, since my first work more than 10 years ago in my lab, I have a lot of different students each year, and at the end have a high intra-laboratory rapid to be maybe near 90% using a positive control as a benchmark. And then after there are some transferability that have been done. There are some work from my lab with 5 other academic laboratory in Europe using standard SOP, and testing results from the different abilities were very, very highly similar. And then an extensive inter-laboratory validation was published a few years ago with 7 different private companies with a lot of chemical, more than 80 different chemicals. And a concordance between companies were near 90%. So I would like to thank you for listening, and open to respond to any question.
Fantastic talk. Thank you so much. We'll take one question. I see one in the Q&A box. What is the scope of in vitro models organ-specific genotoxicity?
At the moment, they are not so much. Normally, GenTox assay are used on classical cell type, like T-cell cells in most of the time, not before macronuclei. But that's a good point, because now we can use these biomarkers that are present in all cell types, all organs, whatever. So that that's part of the work I've done at the moment, where I'm working with different cell types from different organs from human. Last paper I've published, I've worked with neuronal cells, liver, colon, and kidney. And clearly you can see that for some type of compound, you do not see the same type of result for genotoxicity. So I think it's a plus to work with more diverse cell type than only one cell type.
Great, thank you. Since we're a couple minutes over, I'll ask that any other questions for Mark be put in the Q&A box. And Mark, if you could kindly monitor that and answer any of those in writing. And thank you again so much. So we'll go ahead and move on to our next presentation on a platform for next generation carcinogenicity assessments from Drs. Chris Barber and Adrian Fowkes, both of LASA Limited.
That's great. Thank you very much. Thanks for the opportunity to present today. So over the last couple of days, we've seen some amazing work and new assays that are being developed to give us new insights into carcinogenicity. One of the challenges that presents is how to use all of this information and integrate it into decision making. Now, I'll talk a bit more in a moment, but IATAs are a solution to addressing that sort of explosion in knowledge. But I want to show you today how in silico tools can support those decisions and how increasingly I think we'll become dependent upon such tools to guide decision making. And so we'll show you today how we have a platform that supports decision making for ICHS1, and we anticipate this will be a means by which ICCVAM guidances gets updated to show the importance of in silico in NAMs. Next slide, please. So just a very brief introduction for us. So if you don't know LASA, we're an educational not-for-profit charity. And today we're going to talk about a decision support tool called CAPTIS. So the simple way of thinking about this is it's a tool that supports IATAs by automating, injecting expert knowledge, data, and supporting decisions. Next slide, please. So I think we've probably all going to agree with the statement that non-animal studies can now provide enough evidence to support a decision about chemical safety. But that comes with some challenges. in order to gain access to mechanistic knowledge, to know what assays are available, to contextualize the data you get from those assays and know the relevance or specificity of an assay, to explicitly address uncertainty, such as conflicting data results or gaps in knowledge or data, and use all of that to make an informed decision that aligns with regulatory guidance. So I put a small slide, a graph together to kind of show you the way we perceive this. So defined approaches are fantastic when there's a limited number of assays, they're well understood, can be trusted, and the problem space is relatively simple. So you can take a fairly rigid approach, run the prescribed assays, and make the decision as in the defined approach. But as the number of assays increases, as we've seen for the last couple of days, defined approaches start to fall apart, especially if you believe in the principle that you should use all available knowledge and data when making a decision. So IATAs are the way forward, but even IATAs by hand will become unmanageable. And we believe that in the end, AI tools will be needed to support decision makers. And that's what we'll talk about today. Next slide, please. So over the last decade, there's fairly universal acceptance that a great way to organize information is using AOPs. So these link together key events from an initiating event to an adverse outcome. So this way of organizing knowledge can also be used to integrate data and structure decisions. Next slide, please. And so today we'll talk about what we believe are the three fundamental pieces that are needed in order to make those decisions. So one is the integration of information. And we just said that that's primarily, we believe, through organizing data and knowledge against adverse outcome pathways. Applying best practice. So this is harvested by talking to experts around the world about how they tackle the decisions, the type of arguments they would use and when they would use them, and explicitly identifying and addressing uncertainty, because uncertainty in data will end up being risk in humans, and being explicit about how much uncertainty there is and whether that's a tolerable level of risk in the final assessment is really important. Next slide, please. So I want to very briefly talk about the method. This is really the method, not of the development of captives, but the application. So if we start from the premise that adverse outcome pathways are a framework to organize knowledge, they effectively describe the mechanistic causes of toxicity. By appending assays onto those pathways, because assays measure outcomes that can be associated with key events, this then allows you to identify assays that are relevant for assessing toxicity against that AOP pathway. Adding data that's measured from those assays then allows us to organize all of what's known for the compound of interest in a context which allows us to understand the relative value, impact, and trustworthiness of each of those pieces of data. So this is really allowing us to understand how to use a weight of evidence approach and how to weigh each piece of evidence. By talking to experts, we can start to piece together what we would define as best practice. In other words, how experts would confront data which is inconsistent or in disagreement, or how to rate one assay against another. And this then allows others to apply the same way of thinking, or indeed apply their own methodology if they have more knowledge or perspective than the experts we've worked with to create those best practice rules. With all that in place, assessments can be made, such as, have I run enough assays to get enough information to make a decision? Have I appropriately addressed conflicting pieces of evidence and decided which one is more trustworthy and relevant for the decision I have to make, which then allows you to make a decision. And that decision may be, we need to run some more tests before we can come to a conclusion. And eventually you want to be able to share that decision with others, defend it if you need to, and that may be internally to colleagues, or it might be externally to a regulator. And a report, of course, is a vehicle by which that can be done. So that's more or less the method of use of CAPTIS. Next slide, please. So we want to go from the theoretical now to the practical and talk about applying it to ICHS1B. Next slide, please. So just in one slide to describe what that is, in case you're not aware, it's a regulatory guidance that allows an assessment to be made about the relevance and value of running a 2-year CARC study to decide on carcinogenic risk. Essentially, you need to make a decision with 3 possible outcomes. You can either decide from the data you have that a CARC study would not add value because it's likely to be carcinogenic, or that it's not worth running because you have enough evidence to say that it is not carcinogenic. Or alternatively, that you don't have enough evidence and actually a CARC study would still add value in understanding human risk. So these are the 3 possible outcomes you have to make with a decision under S1B. Next slide, please. So CAPTIS has been designed, tested, and applied against this regulatory guidance. So it's aligned to that guidance there. It provides access to knowledge, primarily in the form of the adverse outcome pathways you'll see a bit more of in a moment, along with expert commentary and links to primary literature in order to understand that context. It guides the human to making a decision. It also provides in silico predictions in the absence of experimental data. In other words, where we have associated assays to a key event and where we have access to data for those assays, we can build a model. And we could use that model to make a prediction for the compound of interest. Now, I wouldn't want to suggest that you can use in silico only to make a regulatory decision, but it can certainly help supplement a regulatory decision. It might identify which assays present the greatest risk of a compound failing, in which case you might want to run it earlier, and it can be used to strengthen a decision. So CAPTIS is a piece of software. The science has been published, and I've listed a few of the publications we've presented over the last few years in the journals. Next slide. So in order to make an assessment under S1B, the requirement is to assess 6 factors. So those factors are on the left-hand side of the bottom of the screen in those colored boxes. These are the factors the guidance says you have to make an assessment against individually. CAPTIS organizes its knowledge and information against AOPs, and those AOPs are aligned to those 6 factors, and that's what's shown on the bottom right of the slide. In terms of decision making, we've worked with industry experts around the world to define best practice, by which we mean where these experts all agree this is the logical approach to take and the approach that they would take, we would identify that so that it supports the user when making their decision. Next slide. So in answer to the question of what regulatory testing does your method address, ICHS1B, this guidance. So here we're providing mechanistic or carcinogenicity knowledge, and that's a combination of public knowledge, private knowledge, and expert interpretation. We're also guiding the decision. We're not making it, we're guiding it by the application to best practice that we've defined through collaborations. And we believe this then helps improve the quality of decision-making, increases the consistency of decision-making, and increases the confidence in the decision that's being made. And that's really important, that any decision is transparent and explicable. And so we've created reporting frameworks which are designed to help regulators and others review, challenge, and assess decisions that are made under this framework. Next slide, please. So at the moment, CAPTIS is focused primarily on ICHS 1B. We just had a great presentation on RECAP, and we're watching with great interest how the approach is evolving, and CAPTIS will be aligned to the next set of publications when they come out, so I can't wait to see those. It's also relevant to a working group within the OECD, IATAs, on non-genotoxic carcinogens. But to be really clear, we don't think this is a tool just to support the decisions under one regulatory guidance. We are also working on other endpoints, such as skin sensitization and DART. Next slide, please. So has it been used for regulatory submissions? Yes. Reports generated by CAPTIS have been used by the pharma industry as part of their submissions under S1B. And what I've put here is on the left-hand side, the screenshot from the guidance document itself, and what is expected against those 6 key areas that need to be assessed, the 6 factors. And on the right, I've shown you a little piece of the CAPTIS report. You can see that the terminology and the approach is directly aligned to ICHS 1. Next slide, please. In terms of mechanistic understanding, AOPs are all about understanding mechanism. They are the mechanism by which toxicity pathways can be articulated. So these are the mechanistic pathways. AOPs themselves allow assays and ultimately the data that's generated within those assays to be contextualized and standardized across all of those different assays. And so at the moment, CAPTIS contains 60 assays. I like to see we've got more work to do because from the last couple of days, there's a few more assays that are being created, which will fit against this same framework. And it will have to continue to evolve as we develop new assays that are relevant for providing information that supports a decision. In terms of carcinogenicity, it's mechanistically pretty well understood, and the pathways within CAPTIS represent all of the known major pathways of cancer. And these are pretty comprehensive now, having been developed internally over the last few years by scientific experts in collaboration externally. Next slide, please. We'll carry on, Adrian.
Yep, can do. Thanks, Chris. Hi, my name is Adrian Folks. I'm just going to kind of continue this presentation just to kind of cover some of the criteria raised by the Modelers Developer Forum, which will look into assess what knowledge we have in CAPTIS and how that is used during decision making. So Chris has highlighted a lot about the theory around how decision support tools can help us to perform complex weights of evidence assessments, and I'm just going to look at kind of how it's actually structured into the application, and Chris has highlighted a lot about our AOP framework. So I've just kind of highlighted one AOP. I'm sure we're all quite familiar with them by now, and it's really great that the community are kind of rallying around one construct to represent biological knowledge to capture mechanisms. So one of the questions kind of raised by the forum is about kind of the use of reference compounds, the knowledge within how the knowledge that's used from them. So reference compounds are kind of critical for the development of AOPs, because to have AOPs and key event relationships, they have to be derived from experimental information, and that's used from reference compounds. So if you next click, please, a bit of animation here. Yeah. So once you've got an AOP and a mechanistic, you can then overlay the assays and then look at the results for reference compounds. So for this compound, ciprofloxacin, we can see its activity in the relevant assays for this well-known AOP, and we can conclude that it's going to be a carcinogenic, and it probably operate through the topoisomerase 2 binding AOP. So next slide, please. So just give more flavor of kind of the level of knowledge within our decision support system. Looking at the different factors, we can then count the number of AOPs that are relevant for each factor. But biology is really complex, and actually one AOP can contribute to a number of the factors that have been mentioned in the S1 guidance, hence some of the larger numbers here compared to the total number of AOPs in our system. And I've also highlighted some sort of well-known targets that are relevant for each factor. Next slide, please. So in addition to kind of reference compounds supporting the knowledge of mechanisms that can lead to cancer, reference compounds also have data associated with them, and these can be used during reasoning and expert review for any predictions. So these reference compounds have been tested in assays, and before we've got 60 assays annotated to our AOP network. There's also another degree of complication here, because it's not just the assays need to be mapped, but the measurements and outputs that the assays provide need to be sort of contextualized onto our AOP framework. So the number of measurements might actually be larger than the number of assays. And again, we've got 17,000 compounds annotated into our decision support tool that can then be queried during an assessment. In addition to assays, we've also got in silico models annotated to our network. So we've got a number of Derek alerts, which are annotated to a number of key events. And therefore, if there's no experimental data available for an assessment, you can still access knowledge through predictions. Next slide, please. So some of the questions raised by the forum kind of are more sort of focusing on kind of how does a new test might compare to the existing laboratory animals. So it's a bit different when thinking about a framework for weight of evidence assessment, but I'm going to sort of think about those considerations and see how that applies to weight of evidence. So when comparing kind of our method to existing laboratory animal methods, I think we've got to accept that non-animal methods can be used to avoid the 2-year CART study, and that's kind of embedded in the S1 guidelines. We know that all assays have their limitations and even strengths, and these strengths and limitations can be captured into decision support tools and then used during the weight of evidence. We also know that no single assay at present can cover all known mechanisms that drive carcinogenicity. We're going to have to use a collection of assays to come up with a conclusion. And CAPTIS is going to have all those assays kind of present and contextualized in an appropriate manner. And to come to a conclusion, we'll also need to integrate and reason between all the data that's generated from these diverse sets of assays. And that also requires knowledge to kind of process this information. And again, CAPTIS can capture that through best practice and encode that into its system. And once decisions have been reached, we know that we've got to kind of communicate that to people within an organization or to other assessors at different organizations. So having robust and transparent decisions through good reporting frameworks is really key. So next, please. And overall, kind of want to kind of really highlight here is that safety decisions are kind of moving away from single kind of observational studies in animals to really a kind of collection of diverse observations across a series of assays. This all needs to kind of be brought together through weight of evidence to support a decision. Next slide, please. So also thinking about how our method compares to the laboratory animals, we can take a sort of statistical perspective and perform some validations. But before I kind of talk about some of the numbers of how our software performs, just want to kind of highlight kind of what we're kind of sort of competing against. And this is the sort of the animal studies that we're using to kind of predict for human carcinogenicity. And there's been a number of publications that highlight the limitations of this assay. And we just pulled out one here that show that the 58% of cases were labeled insufficient for classification of human carcinogenicity by EPA. When we look at in silico systems against a human relevant validation set, Direct Nexus is a set of SCR alerts and it has a reasonable sensitivity. But with CAPTIS, a decision support tool, where we're integrating more in vitro data, we can actually increase sensitivity against this validation set. But I just want to highlight here that 85%, this is just through automated reasoning only, and actually no expert has reviewed the data to come to the conclusion, so that number would probably go up under expert review. Next slide, please. So continuing just to look at kind of some technical characterization of our framework through the lens of the questions we might ask about new tests. In terms of variability, decision support tools kind of reduce variability because you're using a common framework that's been expert curated, and that kind of supports kind of reaching confident and transparent decisions. For robustness, within CAPTIS, we've got consistent rules so that evidence is always handled in the same way, through uniform reasoning, and data is handled in the same way as well. In terms of intra-lab reproducibility, I guess from our experience, we've got a number of members using this tool to kind of reach consistent conclusions, not only internally, but getting harmonization with external reviewers as well. And that's really supported by the transferability of kind of the reports we create. We're kind of really supporting those communications between safety assessors at different organizations. Next slide, please. Right, I'm going to hand back over to Chris for some conclusions.
So this is really a bit of a summary slide, where if we walk our way through this slide, S1B allows for the assessment of carcinogenicity of a novel compound, and whether an animal study can be waived, and setting out the requirements in terms of what studies can be done in order to generate enough data is supported by CAPTIS. Once that information is available, it can be organized against that framework of AOPs to contextualize each assays and assess their strengths and weaknesses, to look for gaps. Have I done enough studies to make a decision? Have I addressed conflicting or contradictory pieces of information in a way that is convincing and will be supportable based upon logical arguments? Have all 6 of those factors been assessed, as is mandated by the guidance? And ideally, has expert review been undertaken following best practice, by which I mean the ways in which collections of experts have joined together to agree this is the right approach? And then have we delivered a report that can support that decision and be used in order to make an assessment by a third party? Next slide, please. So just to be clear about outcomes we're looking for today, we wanted to highlight the growing importance of decision support systems. And we really want to open our doors to say that we would like to work with anyone on defining how to assess, adopt, and trust such AI tools. We're part of the way through a number of publications. We'll be happy to have some co-authors from the panel or wider to lend weight to some of these perspectives. Also very interested to know from a regulatory perspective whether you see gaps or needs that aren't yet met. And as an organization, we're highly collaborative and are seeking to work with others to bring into reality the future, which is more and more AI tools will support and guide human making decisions. Thank you very much.
Wonderful. Thank you both. I see that Iris Camacho Ramos has their hand up. So Iris, you should receive a notification on your screen that asks you to unmute your microphone. If you want to unmute yourself and ask your question.
Can you hear me?
Yes.
Thank you, Chris. What analysis have you conducted to evaluate the alignment of CAPTIS with other regulatory frameworks beyond the ICH guidance? Like, for example, alignment to the US FDA, EPA guidance on carcinogenic materials?
We're continuing to seek to align to all the regulatory guidance. Our focus has been on S1. We are looking, for example, at RECAP because we see a very good overlap there. Well, RECAP is based upon the pharmaceutical S1 guidance document. And yes, so we are continuing to work to align to regulatory guidance documents. We see this as critical in decision making.
Thank you.
All Right, there's another question in the chat. The US EPA is using transcriptomics-based studies to develop a RFD used in risk assessments, and others are using this approach for input into OECD, mode of action, risk assessments, and IVIVE. Is this being incorporated into the weight of evidence tool you're discussing today?
Short answer, yes, in the sense that transcriptomics is another means to measure the effects of a chemical in a biological system. And so it will align to mechanisms of action and will align to the AOP framework. So yes.
All right, I'm not seeing any other questions and we are perfectly on time. So we're going to go ahead and take a 5-minute break. Thank you both, Chris and Adrian, for presenting today, and we'll see you all in 5 minutes. All right, welcome back. It is my pleasure to introduce our very last presentation of our very first Method Developers Forum. Today we have Dr. Connie Mitchell from the Health and Environmental Sciences Institute, or HESI, co-presenting with Dr. Jesse Salk of Green Umber. And they are going to tell us about error-corrected sequencing for clonal expansion. So I'll turn it over to you.
Great. Amber, can you just confirm you see and hear me okay? Good. Okay, great. So thanks everyone for this opportunity to speak today. I'm Connie Mitchell. As Amber mentioned, I'm at HESI. I'm really lucky to be joined today by Dr. Jesse Salk. Jesse, would you like to introduce yourself?
Yeah, absolutely. So I'm a medical oncologist, not a toxicologist, and I've spent the last number of years at a company I started focused on error-corrected sequencing for a variety of applications, but I do both human medicine and molecular biology. Worked with HESI for a long time. Great.
Thank you very much, Jesse. Today, we're going to be speaking about the use of error-corrected sequencing for clonal expansion. And this talk is very complementary to other talks we heard yesterday and today, and specifically to Dr. Barbara Parsons' talk on Carcinogenicity yesterday. So this will really complement that. I also want to add that I'm speaking on behalf of a cross-sector group of experts today, made up of our error-corrected sequencing work group. This is led by Dr. Patricia Escobar from Merck and Dr. Megan Myers from FDA and CTR, but there's many folks involved from industry, government, and academia. So I'll go through some of the background and introduction. Jesse will go over the method, and Jesse, please feel free to add as I go, and then I'll cover the context and current use. So next slide, please. So that just says background, so next slide. So I'm sure I don't have to tell this audience too much about cancer progression, but just to emphasize, it's a multi-stage process. As we've talked about and heard in talks today, tools can be used to target different steps along the process. And we have another project I'll mention, but won't go into great detail, that's looking really on that early side for that early mechanistic events like AHR activation. So those tools and other tools we heard about could be paired with other tools that we'll focus on now, including targeting for key events like clonal expansion. And just really emphasizing, as others have done, the use of these data in a weight of evidence manner. One challenge I will highlight is with a lot of these tools is figuring out the thresholds for initiation or promotion leading to tumors. So when do these tools give us enough signal that we think they will lead to tumors, or when are we confident that they won't? So just to emphasize that point. Next slide, please. And this maps out where we see clonal expansion in cancer driver genes along an AOP framework. Again, highlighting the tools from the other project that we have going on at HESI, but other tools mentioned today, really figuring out where these fit in. So we see these as kind of later key events. So next slide, please. Next slide. Oh, there we go. So our project, others have mentioned how this fits in, but we see error-corrected sequencing for clonal expansion to be able to determine the carcinogenic potential of a test agent. And this could fit into regulatory paradigms that we've heard about, like the ICH S1B R1 update, We also heard from Gina and Amber on recap. So for agrochemicals and potentially with the use of industrial chemicals that might have in vivo studies. So next slide, please. So here's the method. So I'll turn over to Jesse for a few slides.
Yeah, so the big picture concept of looking at clonal expansions is really about measuring a somewhat of an aggregated phenotype. So the abnormal ability of cells to proliferate in a way that's atypical. And with a cancer, late stage cancer, we see a large macroscopic mass of cells that look very dysplastic. But early on in the process, cells begin growing a little bit out sort of an abnormal hierarchy of their typical stem cells and may not be histologically appreciable. So the concept is to use a method to track lineage of cells based on tracking mutations that drive proliferation of those cells in a way that we can infer the presence of clones, not based on how they look morphologically, but based on the fact that there is a population of cells that share a particular mutation, which implies that those cells have grown in an abnormal way, and using an approach that can identify, again, these sort of growth changes that happen prior to morphologic changes that we typically see in pathology, with the idea that we could identify processes at earlier stages and be able to do shorter term studies, get information more quickly and more completely. To be able to do this using this concept, we need to be able to measure those mutations with a high degree of accuracy, and being able to measure mutations in a population of cells that are in a small minority. So a group of 100 cells that have a mutation that is clonally expanded, but maybe mixed in with 1 million cells. And the challenge with that is typical traditional DNA sequencing methods have a very hard time in terms of their signal to noise ratio based on the background error rate of the sequencing process, that mistakes overwhelm the signal when you're looking at things at 1%, 0.1%, things really below about 5%. And most DNA sequencing techniques are really indicated for looking at clonal populations, the difference between person A and person B, or something that's in all the tumor cells versus all normal. So to overcome that, make it possible to detect low frequency mutations, we need to use what are called error-corrected sequencing techniques. These are biochemical methods that allow us to drop error rate of sequencing by doing some special biochemical and informatic techniques that make existing DNA sequencers operate at a level where errors are reduced between 1 in 10,000, 1 in 100,000, 1 in a billion. Duplex sequencing is one such error-corrected sequencing method. It's probably one of the most accurate. It's something I developed a number of years ago with colleagues at the University of Washington, and I'll show you in a second how it works. But essentially, it involves using special barcodes in those molecules to create to sequence multiple redundant copies to kind of normalize out the errors. And like I say, the idea is using high accuracy sequencing to detect low frequency mutations that reflect abnormal proliferation of cells, which is itself a biomarker of a carcinogenic phenotype. Carcinogenesis means creation of a process of moving on the process towards cancer, and that's we're trying to do that very early, with the idea being that we can identify these processes on the 3 to 6 month timeline, for example, rather than a traditional 2-year CAR-C study where you're looking for macroscopic tumors. The next slide. The duplex sequencing method involves taking large numbers of molecules that we extract from a tissue, DNA molecules, putting unique barcodes on those molecules so that every single molecule gets a kind of effectively a different fingerprint, and then making lots of copies of those molecules and then sequencing those copies. And the idea is you can look at all the sequence reads that come off and look at the barcodes and then group all the sequence reads together by things that have that same unique barcode, that same fingerprint. And that lets us say, okay, we have, let's say 10 copies that have the same barcode, 5 that came from the top strand of a particular double-stranded DNA molecule, and 5 that came from the bottom strand. And we can look and we can say, in this diagram here, you look in the second column, I'm just showing you an example for one molecule. The top 5 molecules all have, or most of them have, a black A at a certain position, and the bottom strand all have a black T at a particular position. And those red bases represent either random errors or errors that happen during the first cycle of amplification for just one of the strands. And we can say, because those changes are not present and as complementary changes at the exact same position, the majority of molecules, they must be errors, and we can subtract those out. And effectively, by doing this, we can drop error rates below 1 in a million or 1 in 10 million. And it lets us vastly reduce that background noise to be able to pick up very faint signals, which is necessary for seeing, again, say 100 molecules with a mutation out of a million, for example. And the process involves a lot of computational sequencing, but the actual sequencing box, the equipment is the same routine thing that is widely available in a lot of labs. Next slide. So in general, DNA sequencing involves taking DNA out of a tissue. So we take a sample. It could be blood cells, it could be liver cells, it could be any tissue in the body, and you chemically use basically soap and enzymes, real standard process, to get rid of the lipids and proteins and whatnot, and just isolate the nucleic acids. Then we again do this process called library preparation, which is gluing, using molecular glue to attach on these adapter molecules that make it possible to do copying of those molecules, as well as that molecular barcode. You make a lot of copies and you form what's called a library that goes on a DNA sequencer, which then runs for a day or so, and then spits out many gigabytes, terabytes of data that are then taken. The error-corrected consensus making is formed to reduce those errors. Those molecules are, using a computer related to the part of the genome that they came from, so they're aligned. And then we compare how the sequence of that error-corrected molecule differs from the reference genome, the sort of standard against which we compare, and then we identify using again computational pipeline, effectively say, we see a mutation in 17 out of 24,000 molecules at this particular position in a known gene that's involved in the cancer process, and we can infer that there is a small clone reflecting an abnormal proliferation of cells. Next slide. So the way we initially did this work for prototype testing of this, we basically said to track a clone, you need to have a mutation, something that's different in those cells compared to all the others. And the places that you can find those mutations are in genes that are recurrently mutated in cancers. Because by virtue of the fact that those mutations allow those cells to grow abnormally. And so one of the projects we started going is in rats. And the way we look through the entire genome, which is 3 billion bases, you say, well, where do we look? We started by saying, well, let's look at known driver genes in cancer from human cancers, the kind of major ones. Let's look at all the published mouse studies that have been done. And there isn't really a whole lot of work on rat cancer driver genes. There's a little bit, but then we actually did some work ourselves, actually doing whole genome sequencing on rat tumors. And based on all the data we found, what we found in some rat tumors, what we found in published literature from mice, and what we found from humans, we took the orthologs in the mouse and in the humans and found the corresponding genes in the rats, and basically built a panel that picked out some of the major cancer driver genes based on these sort of 3 types of data to come up with a panel that's about 50,000 bases, which is sort of a practical size that's cost effective to work with. Next slide. We did something similar with mice. We looked at the mouse driver genes, human orthologs, and some of the things we learned from the rats, and then picked the mouse version of those. So we created 2 panels, one for mice, one for humans, that are, roughly 30 to 50,000 bases. Next slide. And I'll hand it back over to Connie.
Great. Thank you so much, Jesse. So next slide, please. We'll talk about the context of use. So here, I've mentioned some, but we see this being used for shorter term studies, for pharmaceuticals and ag chems, some of the already required 3 to 6 month studies, and the ability to provide mechanistic information. Another promising part is that we see this being used for both genotoxic and non-genotoxic chemicals. We've heard today about a lot of different assays that can be used for gene tox. For non-genotoxic, it's a little bit more sparse, so that's very promising to be able to have tools that can cover both mechanisms. So that could be used in the system since we're looking later in the AOP. And as we mentioned, it's been mentioned by Barbara, and just to reemphasize here that as far as we know, error-corrected sequencing technologies haven't been used for regulatory submissions to date. It's something, we hope that changes in the future. It's also something that, potentially could be used kind of later on by companies or researchers in getting a compound to market. Again, some of those already required studies trying to use it kind of in-house. So that's where we see it being used. So next slide. We can talk about the 3 Rs. So we see this as being a reduction. So if you find you don't need to do a 2-year bioassay, that reduces the number of animals needed to be used. You still probably have to do the early studies. It's also a refinement because you're using shorter tests, you're utilizing those existing studies, but also you're then getting that mechanistic information. And the long-term goal of using this, and like I've emphasized, other tools could be for the replacement of the 2-year bioassay. So that's how the 3 Rs fit in. So next slide, please. So we'll talk about biological relevance for a few slides. So next slide. So just highlighting the AOP again, really that later stage is where we're focused on. So it can cover, a lot of different molecular initiating events that might converge and other key events that might converge to that. Next slide, please. So the panel had asked about reference compounds. So this is very early days for this. It's only been tested with a handful of chemicals so far across different technologies. So we can't recommend specific compounds at this moment. We really need to do more to establish the model for usage. And to do this, we need to do it with chemicals that have known tumor outcomes in rats or mice. So to do this, we would want to refine the panel and look at organ specificity, and that needs to be done with more chemicals. We would recommend looking at non-genotoxic carcinogens. Again, as I said, there's many tools for genotoxic compounds already. So what we've done in our HESI committee is we've identified non-genotoxic carcinogens that lead to tumors, mostly in rats. There's also info sources that says what it does in mice. And this is really done with experts kind of annotating for the non-genotoxic mechanisms, but also using the public databases like LAZA, and also using from the US government databases of tumor data in rats and mice. So from that, we were able to get a list of potential compounds we could use. We also have an inventory of samples we could use for error-corrected sequencing. We asked our members to look in their freezers what studies have been done that we could potentially do error-corrected sequencing on using this panel of cancer driver genes to try and really establish the method. So I'll talk a little bit more about where we are later, but we do have an inventory of samples we could use to, again, really try and establish the tools. In the future, our preliminary recommendation is that if a lab was trying to get set up to use these methods, they'd probably have to do at least 2 compounds to show their capability. But again, this is pretty early days, so that's our initial recommendation, but need to think about that. It'd be harder to ask a lab to run, 10 compounds. For in vitro, that's a little easier. For these in vivo studies, it's still very time consuming. So that's all I have here. So next slide, please. So now talking about comparing to existing lab methods and animals. So this is a refinement and reduction potentially. It's still using rodent models. We do think it's relevant to human biology though, So what we're doing here is measuring changes in endogenous mammalian genes that are also cancer driver genes in humans. So even though it's still working in the rodent models, we see a lot of, ties to humans and making it human relevant. So next slide, please. So I'll talk a little bit about technical characterization, technical characterizations on the next few slides. So next slide, please. So the panel had asked about robustness. So for error-corrected sequencing, specifically duplex sequencing, there has been a good amount of usage, but not on the cancer driver gene side, as we've been discussing, more for mutagenicity. So it's a different set of probes, it's different genes it's targeting. However, the technical setup for laboratories is the same. Jesse went through and talked about the DNA extractions, library prep kits, and sequencing. So in another HESI committee, our genetic tox technical committee, there has been a multi-site study that's highlighted, that's shown here, the different sites to show reproducibility and transferability. So this paper's in prep right now. It'll come up in the next few months, hopefully, but it really shows that other labs are able to perform the duplex-seq library prep kits and the other steps needed to do this work. So that'll be really transferable for these other probes for cancer driver genes. So next slide, please. So just to emphasize that from this study, again, it's different genes that are being measured. It's the mutagenicity panel, but this will help show the sensitivity, specificity, accuracy, precision, and reproducibility of the method at multiple sites. Again, emphasizing that the setup will be the same for the cancer driver genes. So that means more labs will be really ready to run these methods when we've established more of some of the foundational questions. Jesse, is there anything, I know you've been more involved with this work, anything you want to add or emphasize for this piece?
I think that on the genotoxicity side, the method has been used quite a lot by multiple labs. It's very reproducible. This will get published soon, and there's probably 10 or 15 publications now out there showing this. Underlying technology is quite robust and reproducible, and high resolution for mutagenesis. For the carcinogenesis application, we think the underlying chemistry, processing of samples will be similarly robust, but the biological question is a bit more complicated. The driver genes in different cancers are in different animals, different tissues, maybe different compounds or time points could turn out to be somewhat different, whereas the mutagenesis assay is really focused on looking at a sort of a sampling of the genome and not focusing on anything that's functionally causing the signal. It's just the sort of consequence of random mutations being introduced by mutagens. The carcinogenic use of the technology is a little bit more biologically complex, and so that's part of why this group, is a little bit further along. Things haven't, as many studies haven't been done. We're enthusiastic about it, but there's more question about the biology and lots more exciting work to be done. But the core chemistry, the core technology, we expect to be similarly easy to do from a workflow perspective.
Great, thank you, Jesse. So next slide, please. So I'm going to conclude my talk by talking about where our group currently is. So next slide, please. So we currently have a pilot study going on. As Jesse raised, there's some additional technical questions that need to be answered before we can run many studies in many different chemistries. So since we're looking at cancer driver genes and clones, we need to think about the samples we're testing. So previous work that's been done using this method had done punches of samples like the liver to try not and dilute the signal of the clones. However, there is a chance that if you take a punch of one part of the organ, you might miss the clone. More people are familiar with taking homogenates of a sample. However, then the worry there is that you might dilute your signal too much with the homogenate and then not be able to detect anything. So to try and answer that, we have this study going on using existing samples that have positive tumor outcomes in mice. The samples come from Syngenta, and we're comparing the results of small punches and homogenates in 90-day samples. So this foundation foundational study will give us insight and allow us to run more chemicals after if we know it's better to go with the punches or if it's better to go with a homogenate. And I just want to highlight here, this is a really nice use of the HESI committee bringing people together. So the kits were purchased using committee funds with a large discount from TwinStrand. The samples were donated by Syngenta. The sample preparation, DNA extractions, and library preps were done at the University of Ottawa by Dr. James Alcaraz. And then sequencing was done by Merck under Dr. Keith Tanis. And data analysis is really going to be a collaborative effort, but with folks from the government, industry, and academia all involved. So even just for this pilot, it's been really nice to highlight the use of people from the different sectors coming together to work on this in this forum. So next slide, please. So I'm going to wrap up. This is kind of a general slide, but it shows our vision for after the pilot. So once we have that technical answer to the punches versus homogenate, we plan to run additional compounds. Like I said, we have an inventory we can work from, and those samples have the known tumor outcomes, so we're able to benchmark with those earlier samples. And this is really, again, exciting that we were able to tap our network to ask people to look in their freezers and try and find samples, so we're not at this point generating new studies on animals. There might be a need to refine the panel. So this additional genes piece, it might be something that certain mechanisms or certain organ types need to be refined, but we're trying it right now. What Jesse walked through, it said V1 on it. So it'd be great if it worked, but there might be some refinement needed there. Whole exome sequencing of tumors might also be able to help with that step, but knowing that once something's reached tumors, it might be later on in the process. So we will need to think about what genes would be appropriate to use there. One important thing I mentioned in the beginning is establishing thresholds. So when do we have enough confidence that measuring these clones will likely lead to tumors? And conversely, when do we think it will not? So the ultimate goal, though there's a lot of work to be done there, is regulatory acceptance. But even prior to that, prior to submissions or using this information, I think there's a lot of work that people could do, companies or even researchers could use to try and better understand the chemicals they're testing and really try and utilize this to get more mechanistic information. So that's what I have. On the next slide is my contact info. I'm happy if Jesse would like to add any concluding remarks before we end.
Yeah, I think it's early days, but it's really exciting. You saw from Barbara Parsons, talk yesterday, just how much signal can be, come out in certain organs, certain tissues, certain systems. And I think it will probably vary across species, across different mechanisms, but that variability doesn't mean this is, a problem. It just means it may be, require a little bit more work and focus to have a few different versions of this approach and concept, which probably can be achieved through a number of different technologies. And so I think that the takeaway is about the concept of using new molecular tools that are high sensitivity to look at abnormal cell phenotypes based on tracking of clonal lineage markers, and whether it's spatial technologies in the future, whether it's other forms of error-corrected sequencing, this is a concept I think that will be a major one in the future, and people should just kind of start keeping that in the back of their mind as a major area that I think we will see blossom over the next decade.
Thanks, Jesse. So there's my contact info. I'm happy to chat more about this and what our team's doing, but then happy to have time for questions if there's time, Amber.
Great, thank you both. That was a very nice presentation. We do have a couple minutes for questions. Actually, let me go with the comment first from William Bisson. Says, following up on the final comment from Jesse, not all mutagens are carcinogens. Looking forward to the next steps and great work. And then another question says great sequencing tool. Is there any cell painting or staining assay that may be done as a comparison?
Maybe I can start it. Oh, yeah, Jesse, do you want to go ahead?
Sure. Yeah. So one the idea is you want to actually visualize these clones, and there are some emerging technologies that will be able to look at mutations in situ, and I think that's probably the closest. But Connie can talk about some of the other cell visualization techniques that can be used for looking at early carcinogenesis, not so much on the clonal tracking, but on other aspects of it. Connie?
Yeah, for cell painting, just mentioned we have another project not talked about today that is looking on the in vitro side and really part of that early screening side that maybe could be fit in to a weight of evidence later. For this, we are really discussing, in vivo. So we'd have to be thoughtful on what would be done there. Would we take, a liver and then try and culture the cells and compare there? So I think there'd need to be some thought, but there might be, kind of orthogonal types of information, like maybe you would do cell painting early on in a screen, and then this could be fit in later, to either show some kind of toxicity or lack thereof.
Thank you.
The reality is science requires a lot of good tools, a lot of different tools, and having multiple orthogonal tools is going to really be the way of the future.
What are your plans to share the data analysis, including evaluated libraries, with the scientific community beyond paper publications?
Yeah, it's something, I mean, we've talked about that looking at thresholds isn't simple, and to have a clear and transparent process is going to be important. And I think making the data sets available would be an important first step to that, so people could do their own analysis. We've talked about making code available for different pieces. We didn't speak about today, but right now we're using an online platform called DNA Nexus to get part of the analysis done. But that really gets you one piece, and then you have to do more to look at the clones. Jesse could probably speak more to this, but really having a transparent process that people can understand, I think, will be really important. So we're not just saying, hey, it's not a clone, don't worry about it, or it is a clone, making sure the scientific community understands.
Mm-hmm. And then also, at least within the HESI consortium, there's always a lot of talk about how much of the sample is going to be used up, how much could be kept in reserve for repeat or a future reproducibility study or a new technology. So that's one of the criteria that's discussed. And so if somebody's really interested in using a different method on the same kind of samples, by all means join the HESI meetings, and everyone's very open to that kind of thing.
All right, thank you. I'm not seeing any other questions right now. Before I turn it over to Nicole Kleinsteuer for closing remarks, I just want to mention, I'm going to put a couple links in the chat for everyone. The first is going to be a link to the Method Developers Forum website, and on that link you will be able to access a lot of the MDF resources. We have several pre-recorded videos from various regulatory and industry stakeholders that they very kindly provided these videos that describe their information needs for NAMs for carcinogenicity. Go check those out. That's also the same website where you will find the recordings of the MDF from yesterday and today once they are available. And the second link is going to be a link, if you haven't already, to subscribe to the NICEATM news, which would be an excellent resource for you to look out for information on future Method Developers Forums. So thank you very much for the opportunity to host this event, and I will turn it over to Nicole.
Thank you so much, Amber. A huge, huge thank you to Amber, without whom this event would not have been possible. And to the entire MDF steering committee and to our AV support team and all of our support folks at Innovative, just really outstanding work to make this happen. As I said yesterday, this is the first in a series, so we want this to be an iterative event. And I personally thought that the last 2 days, yesterday and today, were absolutely outstanding. We just within the last 10 minutes, for the very first time, dipped below 100 attendees, but I think that's because people have other stuff they have to go do. But essentially throughout the course of the 2 days, we were holding steady between 100 and 150 people. So really robust turnout, really amazing presentations. One of the things that I particularly enjoyed was seeing some of the points of connectivity between the different presentations. So one of our objectives here, of course, was to increase awareness and create a forum for dialogue between method developers and end users. But I think also there are a lot of opportunities for connection and collaboration among the different method developers and among the different groups that have presented over the last 2 days. So I'm really excited to see where that leads. So that's it for me. Thank you so much to everyone for taking time out of your busy schedules. Thank you in particular to the method developers for presenting your outstanding work. And everyone stay tuned. You know, this recording will be posted within the next couple of weeks, and then we're going to start coordinating within ICCVAM to identify the next topic for our next Method Developers Forum, and you'll start hearing about that in the coming months. So thanks, everyone. Enjoy the rest of your day.